stay close to the mic, that would be helpful.
Now, by far the most well-developed body of theory we have for understanding male reproductive allocation is sperm competition theory. And it's important to note that although a few of these models were designed with the hypothetical external fertilizer in mind, they make a lot of assumptions that don't necessarily make sense for that group that are better suited to internal fertilizers. And in particular, the assumption of complete fertilization, that in any given mating event, a male can completely fertilize the eggs of a female. Nevertheless, the predictions from this huge body of theory are really pretty consistent. With increasing sperm competition, we expect an increase in ejaculate expenditure. And within species, we expect a facultative adjustment for males to more adequately compete against their rivals. That is, for example, allocating more ejaculate to matings with females who've already made non-virgin females. But it's unclear how relevant these predictions may be for broadcast spawners. And part of the reason why we wonder about this is there are a few pretty strange empirical patterns that we have that don't really jive with these sperm competition predictions. In particular, across a huge diversity of taxa, broadcast spawning males seem to release sperm very slowly over long periods of time. They release sperm earlier and longer than females. And this is a little bit unusual if you're thinking about sperm competition with approximate male rivals, where you might want to numerically dominate by releasing sperm quickly and increasing the local sperm concentrations. We can also get some pretty bizarre allocation patterns where large males release about the same amount of sperm as small males in a given mating event, even though they clearly have the resources to outcompete them. And these patterns really beg an explanation because external fertilizers have massive gonads. They are expending a huge amount of energy towards reproduction, dwarfing even the most extravagant internal fertilizers. So what's going on here? Well, a lot of things are going on. Sex is very different for external fertilizers than it is for internal fertilizers. By releasing gametes into the external environment, the ejaculates are exposed to natural selection in that environment. They diffuse rapidly as sperm advects away from males, depending on the local hydrodynamic conditions. We suspect sperm wastage can be really important in this group. There's also this really fascinating density-dependent phenomenon of polyspermy, where because of a time lag between initial fertilization and when the permanent polyspermy block takes effect, there's a time window where eggs can be fertilized by multiple sperm, which they say mortality. And this results in this really characteristic density-dependent fertilization curve, where with increasing density or sperm concentrations, fertilization success increases up to a threshold before polyspermy starts happening more frequently and fertilization success plummets. So the name of the game for broadcast spawning males is really about trying to optimize these local sperm concentrations. And to do that, one of the most important traits that can be under selection are phenological traits. That is, the timing, the duration, and the rate at which sperm is being released. So what are the consequences for male phenologies? Well, if we return to our sperm competition theory and modify some of our assumptions to suit broadcast spawners, allowing incomplete fertilization and the possibility of polyspermy, we get some pretty different predictions. So in the top panel here, you can see the gray line represents the predictions from a standard sperm competition intensity model, and the other lines in the plot show model predictions that have been modified for broadcast spawners. And what you see is this really striking difference that with increasing sperm competition, we see that males are predicted to release less and less sperm in any given mating. In the bottom panel, you can see, if you focus on the black line, we've also shown that, theoretically, we should expect the evolution of greater phenological variances for males with increasing sperm competition, again, releasing sperm over long periods of time here. On the empirical side of things, there have been some really great work looking at sperm competitive trials and induced spawning experiments, which have shown that males should be spawning before females, and that there are interesting trade-offs for how early you should be spawning, with really early spawners having a competitive advantage to fertilize females' eggs from distant females, but at the cost of competing more effectively for more proximate females. Now, this is all in the competitive context, but I mentioned earlier that these ejaculates are subject to natural selection from the environment, independent of what's going on in terms of sperm competition. So what's happening here just in the process of sperm dispersal after sperm is released until it makes contact with eggs? Well, we don't have a whole lot to go on here. There was a pretty good study done a few years ago that manipulated sperm release rate in a flume and showed that when sperm is being released slowly, a second batch of eggs down downstream, he actually receives higher fertilization success. And this is really where the project I'm working on is trying to kind of make a contribution. I want to explore more diverse reproductive phenotypes here. And in particular, I want to incorporate male allocation here. How much sperm should males be releasing and how quickly? And to do this, I used the very majestic and charismatic marine tube worm, Galeolaria cespitosa. And Galeolaria is not just attractive as species, they also have this really nice feature that their eggs are really negatively buoyant, which means in the context of a flume experiment, where I can put them in a flume and find them again. And it also means that we don't lose a tremendous amount of realism relative to what happens in the real world, where when females spawn eggs, they tend to stay in, kind of embedded in the substratum near where they respond. So I'm going to tell you about two experiments I ran recently in this large flow-through flume that you can see in the picture. The first one is essentially a pilot experiment to make sure that the flume was working the way I thought it was working. And I basically manipulated the amount of sperm being released down the flume and measured fertilization success at a patch of eggs downstream. The second experiment, I was looking at phenology. And in particular, I was crossing the total amount of sperm being released with the rate at which it was being released downstream. And in this experiment, I had two patches of eggs downstream, similar to that experiment I described earlier. Now, just to go into a little bit more detail about how I designed these ejaculates. Galeolaria is pretty small, so with individual ejaculates doesn't give you much to work with. So what I ended up doing was I pooled sperm from about 10 to 15 males, and then I diluted it and created aliquots for treatments at 1, 2, and 3 times the average male allocation. And then I kind of harness natural variation in the raw concentrations from these pooled ejaculates to kind of, among runs, sample up more of the kind of allocation parameter space. I release sperm either fast or slow in 1 10-second burst or in 4 10-second bursts spread out over 4 minutes. And I had 2 egg patches that I included as a variable. So for those of you who are not used to or not familiar with flume studies, this is just a really quick schematic of basically what it looks like. So we've got water flowing from left to right. We've got our 2 egg patches here. I'm releasing sperm, the ejaculate, from the syringe at the head of the flume. Sperm is diffusing down, flowing out the back. It's non-recirculating, so the sperm only gets 1 crack at the eggs, which I then collect and later on score for being fertilized, unfertilized, or polyspermic. And for this experiment, I analyzed fertilization rate as moved by domain variable using Linearist and Bayesian hierarchical framework. So the results from the first experiment were pretty simple, but also pretty encouraging. Most importantly, they confirmed I was on the left-hand side of that fertilization curve I showed you before, which is really important. I'm not dipping down into severe polyspermy. If that happens, you really have to take a different modeling approach because you have to be able to account for the fertilization kinetics of polyspermy here. But for now, we're kind of happy with it. It was also nice because it just showed that although I had some, I didn't have really dramatic among run variation here. The results from the flume were pretty repeatable. From the second experiment, the big result was a significant interaction between the amount of sperm being released down the flume and the rate at which it was being released. And so you can see in the figure, the red line is slow release and the blue line is fast. And so the big result here being that at low allocation, or when very little sperm is being released down the flume, highest fertilization success occurs when the sperm is being released very slowly. In contrast, when a lot of sperm is being released down the flume, highest fertilization success happens when release rate is fast. And just to give you an idea of the significance here, after accounting for among-run variation, I'm showing you just the densities, the posterior densities for the simple contrast between these regressions at the high and low sperm numbers. So we can kind of tease this effect apart a little bit by looking at the egg position effect. And in particular, what I want to draw your attention to is the comparison between the 2 blue lines. In particular, that we had steeper gains in fertilization success in the distal egg patch, that is the dotted blue line, relative to the near egg patch. And that's really interesting because it's the complete opposite result of what we got in that previous study, which really kind of highlights how it's really important that the optimal release rate really depends on how much sperm is being released down the flume. Now, there's another way to visualize this data, which maybe is more intuitive for some people and also kind of brings up some interesting questions. And that's if we just do a simple transformation. These are not new regressions. I've just transformed the y-axis to per gamete fertilization rate. And the pattern that jumps out right away is this really, really dramatic decline with increasing sperm allocation. And if we take these together, we first one right away is clearly sperm wastage is a big problem. Most importantly to me, we have this really interesting result that, if you remember, these fertilization curves are sigmoidal on the left-hand side here, which means males can achieve this most rapid increase in fertilization success at the inflection point. And what we would normally consider high fertilization success is past that inflection point. And yet that corresponds to really, really low per gamete fertilization rate. This means males are paying a really, really steep ante just to play the game up where they can actually achieve high fertilization rates. And I think that's just in itself a great explanation for why we're seeing some massive allocation to gametes and gonads in this species. But again, at any given allocation, we expect males to try and maximize their per gamete returns. It comes back to the optimal rate depending on how much sperm is being released. Again, slow males should be experiencing selection to release sperm, small males or males releasing very little sperm should be experiencing selection to release it slowly, while large males or males releasing lots of sperm should be releasing it quickly. But this is kind of an interesting scenario if we recall the predictions from those sperm competition models, which are saying that we should be releasing sperm slowly if you're competing with a lot of other males. I think this is interesting because it sets up a neat scenario where for small males releasing very little sperm, these two processes might be generating selection in the same direction to release sperm slowly. Whereas large males or males releasing a lot of sperm might be experiencing opposing selection here, where if all they care about is just achieving fertilizations in the absence of competition, they should be releasing sperm fast. But in order to more effectively compete against other males, they should be releasing sperm slow, which I think is a really kind of neat and counterintuitive result. And with that, I guess I will wrap it up and take this.
I guess I started thinking while you were talking about a slightly different angle on this, but it seems like there ought to be a lot of density dependence in this. If the density of the species is high, sperm wastage should be less of an issue, I would think. And if it's very low density, sperm wastage would become such an issue that maybe the species could no longer persist because they would have to invest too much in sperm. Is that the case?
Certainly, although remember, density dependence here gets really complicated, especially once you hit densities where you start getting polyspermy, because you then start getting really strong constructive selection.
Yeah.
Yes, absolutely.
Huh. But you didn't
No,
manipulate
I'm not
density in
density
your...
in these.
Yeah.
It's pretty hard to do. Yeah.
Are males aware of the strategies of other males? And do they update?
Sorry, I can't.
Are males aware of the strategies of the males around them? And do they update their strategies?
So certainly males can sense when other males around them are spawning. In fact, there's one of the ways we can actually induce spawning for these guys. If we kind of give them a whiff of sperm, we can actually get them to spawn. So, but it actually seems to be more like a trigger for them to actually start competing. They sense the game's on than anything more sensitive. All right. Well, thanks for coming, both to my talk and also to the session.